Hi viewers, welcome back to one more other lecture session from this channel. So in this session we will be studying regarding biomolecules. So here what are biomolecules and how the specific biomolecules they play an important role for the sustenance of life on the surface of the earth. So in nature here we can observe many of the living organisms, and these living organisms they may include plants, animals and even the microbes. So here all the living organisms they are composed of certain organic elements. So here in order to trace out the specific organic elements which are present in case of the living tissue, here we can take the advantage of elemental analysis. Is it clear? So here by taking the advantage of elemental analysis, here we can trace out the specific organic elements which are present in case of the living organism. So here most commonly the specific organic elements which are present in case of the living organism, it may include carbon, hydrogen and as well as oxygen. Is it clear? So let me write it on the board. Living organisms are composed of certain organic elements. Right? And these include carbon, hydrogen and as well as oxygen. Then in the same way, if we perform the similar observation in case of the non-living component like earth crust, and again here we observe the similar organic elements like carbon, hydrogen and as well as oxygen, the specific organic elements which are present in case of the living tissue, the similar organic elements even they are observed in case of the non-living component like earth crust. Is it clear? Non-living component contains similar organic elements. And again these may include carbon, hydrogen and as well as oxygen. Then what is the basic difference in between the living component and as well as the non-living component? So here both in case of the living component and as well as the non-living component, the specific organic elements which are present they are very similar. Then here what is the basic difference in between the living component and as well as the non-living component? So here the variation it relays, or here the variation it is observed only in case of the concentration. In case of the living organisms, the concentration of these organic elements it remains at higher proportion, but whereas in case of the non-living component the concentration of these organic elements it remains at lower proportion. Is it clear? Living organisms contains higher proportion of organic elements. Right? And then after, if we look at the analysis of chemical composition, how we can analyze the chemical composition of a living tissue. So here in order to analyze the chemical composition of a living tissue, here we should consider one particular living tissue, right? It may be a plant tissue or animal tissue. Analysis of chemical composition. So here in order to analyze the chemical composition in case of the living tissue, here we can take the advantage of one particular living tissue. It may be a plant or animal. So here after considering the particular living tissue, let us assume here the living tissue is plant tissue, right? So here after considering the particular plant tissue, here we should degrade the particular plant tissue by taking the advantage of mortar and pestle. So here after degrading the particular plant tissue by taking the advantage of mortar and pestle into multiple pieces, here we should subject a specific solution which is nothing but trichloroacetic acid solution, which is having a chemical formula Cl3CCOOH. Trichloroacetic acid solution, right? Trichloroacetic acid solution, which is having the chemical formula Cl3CCOOH. So once when we subject the trichloroacetic acid solution to the degraded plant tissue, here we can obtain a slurry-like appearance. So here the particular slurry-like appearance which has obtained after subjecting the trichloroacetic acid solution, basically it contains two specific components. So here in order to differentiate the slurry-like appearance into two specific components, here we can take the advantage of chemise cloth or cotton cloth. So here by taking the advantage of chemise cloth or cotton cloth, here we can differentiate the slurry-like appearance into two specific components, and these include acid soluble pool, or in general terms here we can consider it as filtrate, and the other one is acid insoluble pool, or in general terms here we can consider it as retenant. Acid soluble pool, acid soluble pool or filtrate, and the other one is acid insoluble pool or retenant. So here scientists they have revealed that, or scientists they have observed that the particular acid soluble pool, it contains many number of organic compounds. Is it clear? The particular acid soluble pool, it contains many number of organic compounds. Organic compounds, right? So here by taking the advantage of certain separation techniques, here we can separate one particular organic compound from the other. Is it clear? The particular acid soluble pool, it contains many number of organic compounds. So here in order to separate one particular organic compound from the other, here we can take the advantage of separation techniques. So here by taking the advantage of separation technique, here we can differentiate, or here we can separate one particular organic compound from the other. Then in the same way, by taking the advantage of chemical analysis, here we can elucidate the structural formula and as well as the chemical formula. Is it clear? By taking the advantage of chemical analysis, here we can elucidate the structural formula and as well as the chemical formula. So here these organic compounds which are having the structural formula and as well as the chemical formula and which are present in case of the living tissue, here they are termed as what? Biomolecules. Is it clear? The particular organic compounds, they has structural formulae and as well as chemical formulae. So here these organic compounds which are having the structural formula and as well as the chemical formula, here they are termed as what? Biomolecules. So here why these specific molecules they are termed as biomolecules? Because these specific molecules, they are responsible for the sustenance of life on the surface of the earth. So here for the sustenance of life on the surface of the earth, these specific biomolecules, they play an important role. So the reason why here these specific molecules, they are termed as what? Biomolecules. So here you may get a small clarification. Does the living tissue, does it only contains the organic compound? Does it won't contain the inorganic compound? So here the answer to this question is no. One particular living tissue, it contains both organic compounds and as well as inorganic compounds. So here in order to conclude that one particular living tissue, it also contains inorganic compound, here we need to perform a simple observation. So here by taking the advantage of this particular simple observation, here we can conclude that one particular living tissue, it not only contains organic compound and in addition even it also contains inorganic compound. Is it clear? So let me arrange the things here. As I have already mentioned you, living tissue or living organism contains both organic compounds and as well as inorganic compounds. Organic and as well as inorganic compounds, right? So here, in order to prove that one particular living organism, it also contains inorganic compound, here we need to perform a simple observation. So here let us consider one particular living tissue, and here the living tissue is nothing but what? The plant leaf. Is it clear? So initially the particular plant leaf, it contains water, so the reason why here it is termed as what? Wet white. But once when we expose the particular plant leaf to the sunlight, here all the water content which is present in case of the plant leaf, it undergoes evaporation. Then after, here we can obtain the dry leaf. Dry leaf, right? So here, once when we expose the particular dry leaf to the fire, or once when we burn the particular dry leaf, so here all the carbon compounds, they get oxidized in the means of carbon dioxide and as well as water vapor. Is it clear? All the carbon compounds which are present in case of the dry leaf, they get oxidized in the means of carbon dioxide and as well as water vapor. Then in the same way, all the organic compounds which are present in case of the particular dry leaf, they are capable to liberate the heat in the presence of oxygen by the means of a specific mechanism which is nothing but what? Combustion. Is it clear? So let me write it here. Carbon compounds gets oxidized in the means of carbon dioxide and as well as water vapor. So here in the presence of fire, right, in the presence of fire, the particular dry leaf which contains all the carbon compounds, they get oxidized in the means of carbon dioxide and as well as water vapor. Then in the same way, all the organic compounds which are present in case of the dry leaf, organic compounds which are present in case of the dry leaf, they are capable to liberate the heat in the presence of oxygen, and this specific phenomenon here it is termed as what? Combustion. Are capable to liberate heat in the presence of oxygen, and this specific phenomenon here it's termed as what? Combustion. And it ultimately here ash is obtained here. Once when all the organic compounds, once when they are capable to liberate the heat in the presence of oxygen by the means of combustion, ultimately here ash is formed. Ash. So here the particular ash which has formed after burning all the organic compounds. So here the particular ash, it contains many number of inorganic elements. Is it clear? The particular ash which has formed, it contains many number of inorganic elements. So here what are the various inorganic elements here? The various inorganic elements include calcium and as well as magnesium, and even the particular ash, it also contains certain inorganic compounds like phosphate and sulfate. Inorganic elements: calcium and magnesium. Calcium and magnesium are present in the form of inorganic elements. Then in the same way, even the particular ash, it contains certain inorganic compounds. Inorganic compounds, what are those? Phosphate and sulfate. Phosphate and sulfate. So with this simple observation, here we can conclude that one particular living tissue not only contains organic compound, and in addition, even it also contains inorganic compound. So here we are much more concerned regarding only the organic compounds, but not about the inorganic compounds. So here these specific organic compounds which are having the structural formula and as well as the chemical formula, from the chemistry point of view, these are having certain functional groups. Is it clear? These particular organic compounds which are present in case of the living tissue, these are having certain structural formulae and as well as the chemical formulae. And from the chemistry point of view, these particular organic compounds, they are having certain functional groups. So here, what are the various functional groups which are present in case of these organic compounds here? The various functional groups, these may include aldehyde group, ketone group, amino group, carboxylic group, and even many number of aromatic compounds are observed. And in addition, heterocyclic carbon compounds are also observed here. Is it clear? So let me write it on the board. From chemistry point of view, these specific organic compounds, they have certain functional groups, and these include aldehyde group, ketone group, amino group, carboxylic group, and in addition, even they also contain certain aromatic compounds. Is it clear? And from the biological point of view, these are termed as what? Biomolecules. From biological point of view, they are termed as biomolecules. And these biomolecules, they may include amino acids, carbohydrates, nucleotides, fatty acids. Is it clear? So here all these are nothing but what? Biomolecules. Is it clear? So at first we will be studying regarding amino acids. So what are amino acids? Amino acids, they are nothing but they are the building blocks of protein. So once when many number of amino acids, once when they undergo aggregation, here it leads to the formation of what? One specific polymeric protein. Is it clear? So let me arrange the things here. And if we consider amino acids, so here what are amino acids? Amino acids, these are the building blocks of protein. Is it clear? If we consider one specific polypeptide chain or protein, it contains many number of amino acids. Once when many number of amino acids, once when they undergo aggregation, here it leads to the formation of one specific polypeptide chain or protein. So the reason why here amino acids, they are termed as what? Building blocks of protein. So let me write it on the board. Amino acids are the building blocks of proteins. And in nature, there are more than 300 amino acids. Though there are more than 300 amino acids, but only 20 amino acids, they are most commonly observed nearly in case of all the biological protein. So the reason why here the 20 amino acids, they are termed as what? Standard amino acids. Is it clear? 20 standard amino acids are present. And if we consider the structure of the amino acid, basically the particular amino acid structure, it has two functional groups, and these include amino group, carboxylic group. And in addition, even the particular amino acid, it contains single hydrogen atom, and in addition, even the specific amino acid, it contains one R group. So let me write it here. Amino acid contains amino group, carboxylic group, hydrogen atom, and as well as one R group. Is it clear? And if you look at the structure of amino acid, easily we can understand. So this is the basic structure of amino acid. Is it clear? So this is the basic structure of amino acid. Basic structure of amino acid. So this is the amino group and this is the carboxyl group, and here it is nothing but what? The R group. And in addition, if you look at here in the basic structure of amino acid, here we can observe one specific carbon atom, and here the specific carbon atom, here it is termed as what? Alpha carbon or chiral carbon or asymmetric carbon. Is it clear? The specific carbon atom which is observed in case of the amino acids, here they are termed as what? Alpha carbon or chiral carbon or asymmetric carbon. So here, why the specific carbon atom, here it is termed as alpha carbon or chiral carbon or asymmetric carbon? Because the specific carbon atom, it is in association with 4 different valency positions. So the reason why here the specific carbon atom, here it is termed as chiral carbon or asymmetric carbon. For suppose if the specific carbon atom, if it is in association with 4 similar valency positions, under such condition the specific carbon atom, here it is termed as what? Achiral carbon or symmetric carbon. But here in case of the amino acid, the specific carbon atom which is present, it is termed as chiral carbon or asymmetric carbon, because the particular carbon atom, it is in association with 4 different valence positions. So the reason why here it is termed as chiral carbon or asymmetric carbon. Amino acid contains alpha carbon or chiral carbon, or even in other terms we can consider it as asymmetric carbon. Asymmetric carbon, right? So nearly in case of all the amino acids, the amino group and as well as the carboxyl group, it remains similar. But here the variation it occurs only in case of the R group. Is it clear? In case of all the 20 amino acids, amino group and as well as the carboxyl group, that means the functional groups, they remain similar. But here the variation it occurs only in case of the R group. So here, in order to understand how the specific R group, it plays an important role for differentiating one particular amino acid from the other, let me give you some common examples. If the particular R group, if it is occupied by the means of a single hydrogen atom, then the specific amino acid, here it is termed as what? Glycine. So look at here. Single hydrogen atom, right? So here it is termed as what? Glycine. Then in the same way, if the specific R group, if it is occupied by the means of methyl group, then the specific amino acid, here it is termed as what? Alanine. 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 Then in the same way, if the specific R group, if it is occupied by the means of hydroxyl group, then the specific amino acid, here it is termed as what? Serine. Serine. Is it clear? And in addition, here we should understand, or here we should know how the specific amino group, carboxyl group and even the R group, they are responsible for regulating the physical properties and as well as the chemical properties of the amino acid. Is it clear? The particular amino group, carboxyl group and even the R group, they are responsible for regulating the physical properties and as well as the chemical properties of the amino acid. So here, in order to understand, let me give you some common examples. So let me arrange the things here. And as I have already mentioned you, amino group, carboxylic group and even the R group, they are responsible for regulating the physical properties and as well as the chemical properties of the amino acids. Carboxylic group and even R group are responsible for regulating physical and as well as the chemical properties. chemical properties of the amino acids. So here, in order to understand this particular concept, let me give you some common examples. So the very first one is polarity. So here, what is polarity? So if we consider one particular biological protein, and if we dissolve the particular biological protein in water, most commonly the specific protein which contains hydrophilic amino acids, they remain at the exterior side, but whereas the hydrophobic amino acids, they remain at the interior side. So here, why the specific hydrophilic amino acids, they remain at the exterior side? Because hydrophilic amino acids, they are water soluble. In order to dissolve in water, the particular hydrophilic amino acids, they are capable to form the favorable interactions with the aqueous environment, but whereas hydrophobic amino acids, they are not water soluble. So the reason why they remain at the interior side. Hydrophilic amino acids, they remain at the exterior side, but whereas hydrophobic amino acids, they remain at the interior side. So here it is because of what? Because of the variation in case of the particular side chain. So let me show it here. Let us we assume it as the biological protein. So here the particular biological protein, it has many number of amino acids. So here always the hydrophilic amino acids, they remain at the exterior side. Hydrophilic amino acids, they remain at the exterior side, but whereas hydrophobic amino acids, they remain at the interior side. So let me write it here. Hydrophilic amino acids remains at the exterior side, but whereas hydrophobic amino acids, they remain at the interior side. Hydrophobic amino acids remains at the interior side. And in addition to this, let me give you one more other common example in order to understand how these specific groups, they are responsible for regulating the physical properties and as well as the chemical properties of the amino acids. So here, as we all know, the secondary structure of the protein, they are of two different types, and these include right-handed alpha helix and the other one is beta pleated sheet. Is it clear? So here it is nothing but what? Right-handed alpha helix. Secondary structure of protein are of two different types, and these include right-handed alpha helix, and here it is nothing but what? The beta pleated sheet. So here all these are nothing but what? The amino acids, right? So here in case of one particular alpha helical turn, here we can observe 3 amino acid residues average. So all these are nothing but what? The amino acid residues. So here the right-handed alpha helix, it is stabilized by the means of intramolecular hydrogen bond. Is it clear? The right-handed alpha helix, is stabilized by intramolecular hydrogen bond. Intramolecular hydrogen bond. And if we look at the particular right-handed alpha helix, most commonly the specific right-handed alpha helix, they are composed of certain amino acids like LACME amino acids. What are those? LACME amino acids. Right-handed alpha helix are composed of LACME amino acids. What are those? LACME. So here L stands for what? Leucine. A stands for alanine. K stands for lysine. M stands for methionine. And here E stands for glutamate. So here they are simply termed as what? LACME amino acids. So here most commonly in order to form the right-handed alpha helix, these specific LACME amino acids, they are responsible. So here in order to fit into the right-handed alpha helix, as we all know the particular right-handed alpha helix, it is a twisted helical like structure. So here in order to fit into the right-handed alpha helix, only these specific amino acids, they are most commonly preferable. Is it clear? It is because of what? It is because of the variation in case of the side chain. Then in the same way, if we consider the beta pleated sheet, most commonly the particular beta pleated sheet, it is composed of certain bulky amino acids. Is it clear? The particular beta pleated sheet, it is composed of bulky amino acids like isoleucine, phenylalanine and tryptophan. Beta pleated sheet is composed of bulky amino acids. And in what way the right-handed alpha helix, it is stabilized by the means of intramolecular hydrogen bond. Then in the same way the particular beta pleated sheet, they are stabilized by the means of intermolecular hydrogen bond. Beta pleated sheet, they are stabilized by the means of intermolecular hydrogen bond. Intermolecular hydrogen bond. So here in order to form the zigzag orientation in case of the beta pleated sheet, these specific bulky amino acids like isoleucine, phenylalanine and as well as tryptophan are responsible. Isoleucine, phenylalanine and as well as tryptophan, they play an important role. And it is because of what? It is because of the variation in case of the side chain. So here all these are nothing but what? These are the examples in order to predict or in order to conclude that here these specific functional groups and even the R group, they are responsible for regulating the physical properties and as well as the chemical properties of the amino acid. And then after here we are going to study how the specific amino group and as well as carboxylic group, they are responsible for differentiating one particular amino acid from the other. Is it clear? Here in case of all the 20 standard amino acids, amino group and as well as carboxylic group, they are similar or they are present. But in case of certain amino acid, here there is an availability of more than one particular carboxylic group. So here the specific amino acids which are having more than one particular carboxylic group, here they are termed as what? Acidic amino acids. Then in the same way, if a specific amino acid, if it is having more than one particular amino group, here they are termed as what? Basic amino acids. So let me show it on the board. So let me arrange the things here. And as I have already mentioned you, if a specific amino acid, if it is having more than one carboxyl group, then the specific amino acid, here it is termed as what? Acidic amino acid. So look at here. Acidic amino acid, right? Acidic amino acid. So here the particular amino acid, it is having more than one carboxylic group, and the best example is aspartic acid. Then in the same way, if a specific amino acid, if it is having more than one amino group, then the specific amino acid, here it is termed as what? Basic amino acid. Basic amino acid. Basic amino acid, and the best example is lysine. Is it clear? And in addition to this, here we should understand one of the remarkable feature in case of the amino acids, which is nothing but ionizable pH. The specific amino acids, they are capable to resist both the acidic pH and as well as alkaline pH. Is it clear? In the presence of acidic pH, here the specific amino acids, they act as proton acceptor. Then in the same way, in the presence of basic pH or alkaline pH, the specific amino acids, they act as proton donor. Is it clear? So let me arrange the things here. As I have already mentioned you, amino acids are capable to resist both the acidic and as well as alkaline pH. Alkaline pH. So let me show it on the board. Is it clear? And here it is nothing but what? In the presence of acidic pH. And here it is nothing but what? Alkaline pH. So here in the presence of acidic pH, amino acids, they act as proton acceptor. Acidic pH, amino acids act as proton acceptor. So look at here. Is it clear? Initially here it is COO minus, but in the presence of acidic pH, when the specific amino acid, when it is in acidic solution, the specific amino acid, it acts as proton acceptor. It accepts one specific proton from the acidic solution. Is it clear? Then in the same way, in the presence of alkaline pH, here the specific amino acid, it acts as proton donor. So let me write it here. In the presence of alkaline pH, amino acids act as proton donor. So look at here. So initially here it is NH3 plus, but in the presence of alkaline pH, the specific amino acid, it donates one specific proton. So the reason why NH3, it successfully modifies into NH2 in the presence of alkaline pH. So the reason why in the presence of alkaline pH, the specific amino acid, they act as proton donor. And the specific organic compounds which are capable to resist both the acidic pH and as well as alkaline pH, here they are termed as what? Ampholates. Is it clear? The specific organic compounds which are capable to resist both the acidic pH and as well as alkaline pH, here they are termed as what? Ampholates. And the specific nature of these organic compounds, here it is termed as what? Amphoteric nature. So let's write it here. Organic compounds which are capable to resist both acidic and as well as alkaline pH. Here they are termed as what? Ampholates. And the specific nature of these organic compounds, here it is termed as what? Amphoteric nature. Is it clear? So this is regarding the introduction of biomolecules. Then in the next upcoming video session, we will be studying regarding lipids, nucleotides, primary and secondary metabolites in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. Thank you.